once again, so in the previous lecture, I think the previous part of model 2, this is where we got to. So in the next one, we are going to look at transcription. Okay. And transcription simply means copying the genetic information on a DNA onto an mRNA or an RNA strand. So we are synthesizing RNA from DNA and copying the genetic information from the DNA to the mRNA. Okay, that's what we mean by transcription. And transcription occurs on only genes. Okay, so in your genome we have coding parts and uncoding parts. Only the gene regions or gene sequences are the coding parts. Okay, so sorry. In certain organisms like this, their genomes are very effective in the sense that they have little to no introns or sequences that do not code for anything. Okay, I would rather not call them junk DNAs because of course we just don't know what they do, but they are not useless. So but in eukaryotic genome, for example, there are so many non-coding genes, right? And notable of them are the introns. Okay, so we have introns and exons. So the exons are the coding regions and the introns are the non-coding regions between the exons. So let's focus on the exons right now. The exons are the coding regions. So we have to find a way of joining these coding regions so that we can continuously transcribe our mRNA from a DNA strand. Okay, so let's assume this diagram over here is a DNA or let's say a chromosome, a strand of a chromosome, right? And these blue spots are the genes. So you can see there's a blue gene 1, 2, 3. There are 3 genes on this strand. Another notable thing about this is you realize this two-stranded molecule. Okay, and if I don't know if I've mentioned this, but either one of the strands, the 2 strands of a DNA can serve as a template for the transcription or synthesis of mRNA. Okay, so either the strand at the top or the strand at the down can be used as a template for mRNA synthesis. So if you are transcribing this gene, let's say in this diagram, for example, in this diagram, for example, the first gene, we are in the transcription of the mRNA, the first strand was used as a template, right? But in the transcription of the last or the third gene, the second strand was used as a template, right? So there is no rule that on the same strand or on the same stretch of DNA, if you are using one strand for the transcription of a particular gene, then neighboring genes should also use the same strand as a template, right? So in this case, if you are using the top strand, it's going to move from 5' to 3', and mostly we indicate the 5' on the left. So we read from like left to right, so 5' to 3'. So if you are reading from the second strand, which is the one at the bottom, the 5' will be on this side because we know the 2 strands of a DNA molecule should be they shouldn't be parallel, right? They are antagonistic. So if the 5' end of the first one is here, the 5' end of that other one will be here. So if this mRNA is read or the gene is read in this way, that other gene that is using the second template will run in the other way. Does that make sense? I hope it does. I'm sure I've talked about what a gene is. A gene is let me just skip this. So in prokaryotic genes, I think I've mentioned this, they have less or no introns. Okay, the introns are the non-coding regions that are between the exons. So most of their chunk of their DNA or their genome are actually exons. Okay, so let's take a typical prokaryotic gene, for example. So from this side to this side is that the gene region. Okay, the green region is the gene region, and mostly genes must be regulated because they are going to be expressed, and there should be regulation mechanisms, kind of switches, okay, that will either enhance or suppress the transcription or the expression of the gene, right? So sorry for that. So mostly genes have a promoter, okay, upstream of where the gene starts. Okay, so upstream is like before the start point of the gene. So promoters, mostly genes have a promoter and they also have a start where the transcription starts, or transcription start point. Okay, transcription start point won't necessarily be where the gene actually starts, because mostly, we have let me not assume so we have start codons and stop codons, and when we are transcribing a gene, we always look for the first start codon. So there might be a case where the start point for the gene will not be the start codon, so we have to read along the gene till we get to the first start codon. So assuming we find the first start codon somewhere here, okay, the whole of this place is not going to be transcribed, okay, until we get to the first start codon, and the start codon is AUG. Okay, so until we get to AUG, where we find a region of an AUG sequence, we are not going to start transcription. Okay, so the region on the gene before we get to the start sequence is called the 5' UTR, 5' untranscribed region. Okay, and as I said, the promoter is a regulator, so there are other promoter binding proteins and other stuff that binds to it to affect transcription, either negatively or positively. Okay, but mostly they are promoting transcription. So anything ahead of the gene is upstream. So if we say upstream something of a gene, that means it comes before the gene, and downstream is after the gene. Okay. Okay, so, and where a stop codon is as well, I think I've established that we have start codon and stop codons. Okay, so the region before the start codon is the region on the gene before the start codon is the 5' UTR, and the region after the first stop codon, okay, on the gene is called the 3' UTR. Okay, so after we transcribe our gene, we get our mRNA, and the mRNA in prokaryotes, we know prokaryotes do not have a nucleus, so everything is in the cytoplasm. The mRNA will be translated then and then into a protein, right? So I think in prokaryotes, if I'm not wrong and if I'm right, transcription and translation co-occur at the same time, or it can co-occur at the same time. Let me put it that way. Okay, because there is no membrane barrier between the processes. Okay, but in eukaryotes it's different, because transcription is in the nucleus and translation is in the cytoplasm. Okay, and there can be instances where different genes that are coding for particular enzymes that regulates a particular pathway, okay, will be grouped under a particular promoter. This is a lot, but I'll make it simpler. So you realize, let's say we have a pathway like glycolysis, and you realize we have numerous enzymes under glycolysis. So let's say we have enzyme 1, enzyme 2, and enzyme 3 in glycolysis, and mostly these enzymes are proteins. So if they are proteins, they have to be coded for by a gene. So enzyme 1 will be coded for by gene 1, enzyme 2 gene 2, and enzyme 3 gene 3. Because they are catalyzing reactions in the same pathway, they will be grouped under a particular promoter, so that they won't, and you say, hey, I'm speaking to you. So it won't be like each one of them is going to be needing their own promoter, right? So they'll be grouped under a particular promoter. Okay, so mostly we call this kind of genes, the genes grouped under a particular promoter, operons. Okay, and a similar one is the lac operon, the lactose operon. I'm not sure if we're talking about that. Yeah. All right, so that's it about operons as well. So let's look at some characteristics of the bacterial gene. Okay, I think I've already mentioned the UTRs, the promoter regions, but besides them, we have other parts of the gene as well. So let's I've mentioned where the promoter is, and we have a transcription factor binding site. So if a typical prokaryotic gene will have a binding site for certain specific proteins. Okay, so when they bind, they also affect the transcription, either the rate of transcription or whether transcription takes place or not. Okay, and we have some consensus sequences like the TT GACA and the Tata box, or the TATAT, right? So I call this the TT GACA, but it's actually negative 35 sequence. So everything upstream of a gene is in the negative region, okay, and everything downstream is in the positive. Okay, so if a sequence is found upstream of the gene, we actually add a negative to it. So if you are, how many nucleotides away from the gene is it? So probably, oh sorry, how many sequences? So the TT GAGA is like 35 sequences, about 15 to 20. So if you count like 35, so negative 35. So 35, negative 35 upstream of the gene is where you are going to find the TT GAGA. Okay, and the Tata box is negative 30. It's called a negative 10 sequence. Okay, and that is it. The Tata box is made of about 5 to 9 bases. Okay, so negative 35 sequence and negative 10 sequences are upstream of the gene. Okay, I'm not sure if you come across a positive one, but if in case you come across a positive sequence, that means they are found downstream of the gene. And we also have translation binding sites. So this one is on the mRNA, okay, for translation to okay. And in prokaryotes there is a name for the translation binding site. In eukaryotes there is a name. In prokaryotes it's called a Schindler organo. In eukaryotes I've forgotten, either the Kozak, I've forgotten, but I'm not sure you're going to need it because it was in third year, right? But in prokaryotes the translation binding site is called a Schindler organo. Okay, and it is 10 base per upstream of the transcription start point. Okay, so so I think this is the dotted line. I don't know if it's showing one or more open reading frames, and I think I've already explained what an open reading frame is. Yeah, I think that's it. So obviously, because we have a start codon, we have a stop codon. So when transcription is ongoing and it comes across a stop codon, there will be signals for it to stop. Okay, and we are synthesizing mRNA, and you get to know that not in prokaryotes, but in eukaryotes, for example, yeah, there will be addition of poly A tail. But in prokaryotes, we don't add poly A tails. There are different ways of terminating transcription. So we just terminate transcription. Okay. So let's look at. Okay. So let's look at. I think I've mentioned transcription. So let's look at the transcription bubble or this is not a replication bubble. Let me call it transcription bubble. There is nothing like that, but where a part of the DNA is opened up for transcription to occur. Okay, and it moves, so it moves along the replication fork. Okay, I'm calling it the transcription bubble. So in this case, you realize one of the strands is being used as a template, another one is not involved. Okay, so the strand being used as a template is called the non-coding strand. Okay, why? Because when you synthesize an mRNA, it is going to be complementary to the template you are using. So assuming, and by the way, I've gotten my pen and my pen back. In the previous video I wasn't able to access this. So realize you have two strands, right? Let me just, I'm coming. Oh, sorry. So what am I doing? So you realize you have two strands, right? You have two strands, red and green, and assuming the green is being used as your template and the red is not being used. So for instance, you are going to synthesize an mRNA and the blue one is your mRNA. In this case, this blue one is going to be the same as this. Okay, because it is complementary to the green one. I don't know if this makes sense. Initially, it was the red and green that were together, and for two strands to be together, that means they are complementary. Okay, so the red and green are complementary, but we split them up. At first, they were one molecule, but because they are complementary, that's why they be split. Okay, so after the unwinding, they are separated, and the green one is used as a template. Okay, so the red is complementary to the green one, and we've used the green to synthesize our mRNA, which is the blue one. Now the blue one, which is the mRNA, will be the same sequence as the non-template strand. Okay, but because this is the same as the mRNA, we call it the coding strand. Okay, it will be the same except the T bases. So because this is DNA, it will have T in them, but because this is RNA, it will have U instead of T. And I think we've talked about this as well. So this will be called the sense strand or the coding strand or the non-template strand. And this one, the green one, is the template strand or non-coding strand or antisense strand. You can slow the video down and take notes. Make sure it doesn't confuse you. I think I've made good. I've explained better. So transcription is actually catalyzed by a polymerase. Okay, and in bacteria or prokaryotes, it's catalyzed by RNA polymerase. Okay, polymerase is an enzyme that polymerizes. I don't know if you know what polymerization is, but it synthesizes a new strand. Polymerization simply means like so you are just, I don't want to say elongating, but you are just creating something. Okay, so polymerizing is bringing something together. Okay, so adding nucleotides till you get a polymer, right? Because the nucleotides are like monomers. Okay, so you are adding them together to get a long strand. That's what polymerization simply means. So a polymerase is something that polymerizes. Okay, so in this case, we are synthesizing an mRNA, and the mRNA strand is the red one over here. Okay, so let's say we are using this strand, the second strand. Okay, so you realize this point has been already synthesized. Okay, and this point will be the 5' end of the mRNA. Okay, so let's answer some questions. Where is the 5' end of the gene? This is the gene. This is the gene. From this side to this side, the place being transcribed is the gene. So where will be the 5' end? Okay, and where will be the 3' end, and also of the mRNA? But one thing you have to take good notice of is always the 5' end of the mRNA is transcribed first. Okay, so this side of the mRNA will be the 5', and the part we are transcribing is going to be the 3'. So the last part that we the last end that we transcribe is going to be the 3' OH end, or the 3' end, let me put that way. Right, so that's it for the transcription. So as I said, in the mRNA, the U is going to replace the T's, right? Okay, this was another illustration. So this is just like an illustration of different genes being transcribed. Okay, so let's look at similar thing in eukaryotes. So in eukaryotes, because we are complex organisms, of course the process is going to be a bit complex but not difficult. Okay, because it involves many other binding factors and stuff. Okay, so as I said, bacteria do not have introns. Okay, or prokaryotes do not have introns, but eukaryotes do. So we have to make sure we get rid of the introns and join the exons before we can proceed or even start transcription. Okay, so the introns are the non-coding regions, as I said. So let's look at this. This is our DNA strand. Realize the DNA has an enhancer or a silencer. This is a regulatory site. Okay, so transcription binding site. So something will bind, and either an enhancer or a silencer will bind here to either enhance or silence the transcription. There's a promoter. You have the proxima and the core subunit. There's a 5' UTR. It's part of the gene, but that place is not transcribed, and this is where the transcription starts. Okay, and you realize there are some white or grayish areas within the gene. The gene is the red part. So those are the introns, and you have to cut them off and join the red parts. The red parts are the exons. Okay, so we have the process of cutting the introns is called splicing. Okay, and splicing is just another one of different modifications done to an mRNA. Okay, either before or after. Okay, so before or after the synthesis of an mRNA or RNA, let me put it that way. So you realize in this case, like this, in the DNA, we where is it? Yes, so we cut the introns. So there are splices. The machinery that does the splicing is called a spliceosome. Okay, so it's going to cut the introns and join the exons, and we'll be looking at how the splicing is done in subsequent videos as well. So let's look at the eukaryotic nuclear gene structure. So I think we know how the gene in prokaryotes looks like. Okay, with the negative 35 and negative 10 sequences, the promoter and stuff, right? So let's look at the same thing for the eukaryote. So we also have I don't know if there's a picture. Unfortunate. So we have upstream regions, okay, and the we have upstream enhancer element, upstream promoter regions. We also have a gene box, sorry, a GC box, and this is negative 90. And you see there's a negative here. That means it is upstream of the gene, right? And we have a cat box, that's CAAT box. That's also negative 75. That means it's upstream of the gene. So the GC box is 20 base pairs long, and the CAT box is 22 base pairs long. We also have a Tata box, okay, just like okay, this wasn't Tata box, like, because the sequences are TAA, like ATTT, we can call them Tata box, but the order of the nucleotides are not the same. So ours is TATA, and in prokaryotes this is TTAAT or something, forgotten, TATAAT, right? And yes, the negative 10 sequence TATAAT. But in eukaryotes we have a Tata region, okay. Well, so yes, this is ours, and that Tata promoter is a negative 30 nucleotide, okay. We also have a spacer DNA, about 14 to 20 nucleotide long. We have a cap site, transcription initiation site. So there are so many regions. So you realize in prokaryote the translation initiation point was called a shined organo, okay. In eukaryotes ours is called the Kozak sequence or Kozak signal. So this is most, I think it's actually a ribosome binding site, okay. We call it translation site, where the mRNA is going to, the small subunit of the ribosome will bind to the mRNA before, when we are assembling the translation machinery, before the other the larger subunit of the, I'm not sure you guys will remember, but that's how the translation machinery is assembled, okay. So the mRNA will be fixed on the small subunit of the ribosome, okay, and the region on the mRNA that the ribosome actually recognizes is called the Kozak signal or Kozak sequence, okay. In prokaryote it's called a shined organo sequence. Please, if you hear any background noise, forgive me, okay. I'm in a hostel and it's pretty crazy here sometimes, so sorry. Unlike prokaryote, when you are terminating our sequence, or when you are terminating transcription, we add poly A tails, in not in all RNAs, but in most mRNAs transcription, right? We add a poly A, like A, okay, and A is called an adenine, okay. So in this case we call it adenylation. So we are adding A's, and you can guess the enzyme that does adenylation. And I don't know if, I'm sure you know what a poly means. So if you say polyadenylation, that means we are adding like numerous A's. So polyadenylation will be catalyzed by adenylase, okay, and deadenylase will be the opposite of that removes A. So I'm sure you know introns, we've mentioned them. So between exons and introns there are junctions, okay. So from what Doc said the last time, we should be expecting we shouldn't be expecting pictures, okay. So we are just going to get like a sequence of maybe a AGT, CT, GT, right? Let me clear this and use so you're going to get a sequence like in the exam. He's going to give us like numerous letters like this, right? And he will just ask us to find or identify where the introns is, okay. So it is up to us to know where the exon intron boundaries are, okay. So in the exon intron boundaries we have the start point, we have a GU and an AG and a branch site. So these are three things to look out for when we are looking for an intron. Okay, so it might be a lot because it might be like so many letters. Okay, so it is up to us to read through and identify these points. The GT side or the GU side is called the donor site. Okay, so if it is a DNA strand, you are looking out for a GT sequence, okay, or a GT nucleotide. If it is an RNA, you're looking for GNU. Okay, that's the donor site. Then a consensus sequence. This one, obviously, you're always going to come across it. UAC, UAAC. So it might be difficult remembering this. So just remember UAC. This is like two UACs, but the second UAC has double A's. So UAC, UAAC. Okay, that's the branch site or the branch point. And the last one is the aging site. That's called the acceptor site. Okay, and mostly it might not come directly as an aging. You might see like 12 pyrimidine nucleotides preceding it, then an aging. Pyrimidines can be T or C, so it can be CC, like 12 of them, then the aging, right? So be looking out for this when you are asked to point out an intron in a cluster of nucleotides. And the exons, obviously there are no particular site that marks the start point of an exon or not. Okay, so far as the introns are removed, all the other remaining sequences are exons and they are going to be joined and transcribed. Okay, but mostly we actually count them or refer to them within the open reading frames. So from where the start codon is to where the stop codon is, right? That will be our exon. Okay, and the start codon always is an AUG codon or AUG sequence. Stop codons, there are three of them: UAA, UGA, and UAG. Okay, I think you guys know everything already, or I've mentioned them already. Let me put it that way. So 3% of human DNA encodes protein. It's not important. So let's look at the RNA polymerases. I think I've said this. If you see, especially in the case of polymerases, okay, if the annotations given to it or are in like Roman numerals, they are actually referring to bacteria or prokaryotic enzymes. But if they are in the Latin, or I don't know if it's Greek, the alpha beta things, if they are in those ones, they are in they are for eukaryotes. Okay, so the RNA polymerases, we have polymerase 1, 2, 3, 4, and 5. Okay, mostly I think I've mentioned this. The 4 and 5 are for translation synthesis. Okay, so mostly we don't employ them. So the ones of focus are the 1, 2, and 3. RNA polymerase 1, polymerase 2, and polymerase 3. Okay. The polymerase 1, what it does is it synthesizes rRNA, the 40S subunit. Okay, if it is in yeast, the same subunit, but it's called a 35S subunit. So polymerase 1 synthesizes rRNA, but you see rRNA, we have the 5S, 40S, and stuff. Okay, but it does not synthesize the 5S rRNA, only the 40S. Okay, so another polymerase will take charge of the 5S, and you come to know which one. Polymerase 2 synthesizes mRNA, okay, and most small nuclear RNA or snRNA and microRNAs. Okay, so in the case of the transcription we just did, this this RNA you are looking at, but this polymerase here is obviously polymerase 2 because we are synthesizing an mRNA. Okay, so this RNA polymerase is RNA polymerase 2. Okay, where is it? And polymerase 3, it caters for tRNAs and also the small subunit or the 5S RNA. Okay, so even though polymerase 1 is in charge for the rRNA, it doesn't synthesize the 5S rRNA. Okay, so RNA polymerase 3 will take charge of that, in addition to the synthesis of tRNAs and other small RNAs found in the nucleus. I said 4 and 5 are not really going to be talked about. So I think Doc wanted us to rethink the definitions for genes, introns, and stuff. Okay, because introns are defined as the non-coding regions of the genome. Okay, but in the course of our discussion, we are going to come across some introns that code for proteins and they use these proteins to cut themselves. Okay, and this goes against the rules, or goes against the definition that we actually have for introns. A gene as well, I think, what do we call a gene? It's often the segment of DNA that encodes a protein. What about a DNA that encodes for other stuff? Okay, but these are not genes. Okay, so we have to redefine our. So this thing is just for your appreciation. So I'm sure you guys. Okay, let me know. I said I won't be assuming anything. So that's it for transcription. Okay, so the next one is translation. Transcription. Let's focus on mRNA, not the other RNAs. Okay, so in transcribing mRNAs, transcription ends with mRNA, which is a strand of RNA with genetic information copied from DNA. Okay, sorry. So in eukaryotes, for example, the mRNA is going to be transported outside the nucleus to the cytoplasm to be translated into a protein. Okay, and that process is called translation. So on the mRNA, the sequences on the mRNA are going to be read by the tRNA. Okay, but they have to come. I don't know what prompted them. I think they just assumed that there should be a coding system, or there should be a way the tRNA reads whatever is on the genetic information on the mRNA. Okay, so they wanted to know how many bases are read at a time. Okay, or how many bases correlate with a particular amino acid. Okay, and we have only 4 bases but 20 amino acids that in human protein or living organism proteins, right? So if you say one base per amino acid, it's not going to be enough for the 4 bases won't be able to code for the 20 amino acids. 2 bases will also be enough because it will end up coding for only 16 amino acids. But when we did 3 bases, that was enough. Okay, so that's how we came about the codons, the triple codon stuff, right? So codons are simply the bases that the tRNAs read, or the bases that codes for a particular amino acid. Okay, so it has to codon is like a triple nucleotide or 3 nucleotides that correlates with a particular amino acid. So in the case of a start codon, you realize we saw AUG. So whenever you come across AUG stands for or codes for methionine, right? So that's what a codon is. So genetic code. So as I said, we have the start codon and the stop codons. Start codon on the DNA is ATG, on RNA is AUG. Stop codons, we have UAA, UAG, UGA. I think we are almost through. These things are not really important. So this, I think, is just showing us that the various, I don't know, what you have to know about transcription and translation. If in our case here we are not looking at the factors and how the ribosomes or the ribosome is assembled and stuff, there's not really much to say about translation. It's pretty simple if you're not looking at those things, right? There's not much to say. But one thing you have to know is about the tRNA that does the reading. Okay, the tRNA, there's something. Okay, before we actually go to the tRNA, let's look at something called codon degeneracy. Okay, so because if we are doing triplet coding, that means there are going to be surplus codons. That's not going. If you are saying like strictly 3 codons, or let's say 3 nucleotides, strictly 3 nucleotides are for 1 amino acid. Okay, if you are doing that, there will be excess of the nucleotide bases. Okay, but all nucleotide bases have to code for something, and that's where the degeneracy comes in. So different codons can code for a single amino acid. Okay, that's what we call the codon degeneracy. Multiple codons coding for the same amino acid. So I think this table will make it a bit better. Hey, Charlie. Okay, so let's take some 3 examples. like proline. Okay, some will say proline, but proline like this has proline has four codons: CCU, CCC, CCA, CCG. Okay, so this is codon degeneracy. But something like methionine has only AUG, and I think is it tyrosine or tryptophan also has a single codon. Okay, UGG. So tryptophan and methionine have only one codon coding for them, but someone like phenylalanine has three, serine has six, if I'm not mistaken. Serine has two here, four here, serine has six. Okay, and some are non-coding. Okay, the stop codons UAG, UGA, and UAA. Where should that be? UAA. Okay, UAA, UAG, and UGA. Those are the stop codons. They don't code for any amino acid. And one thing you should really take note of is the alphabet for the 20 amino acids. Okay, so we know W is tryptophan, and yes, W is tryptophan, and F is phenylalanine, lysine is K. So you have to know them. Okay, so mostly guys is questions when they come, if he's actually going to talk about a particular amino acid, he might try doing this. Okay, ABA, K residue of particular protein. So you should know K is a lysine residue. Okay, serine, in some case I think serine is S, so that's straightforward. Y is tyrosine, E is glutamate. Okay, and J is glycine. So you can see glutamate and glycine, they are all genes. Like they all start with genes, but which one actually has the alphabet gene? It is glycine. Glutamate or glutamic acid is E, and glutamine is Q. Okay, cysteine, aspartate, asparagine, arginine, and alanine, they all start with an A but different alphabet. Alanine is the E, arginine is R, right? And asparagine is N, aspartate or aspartic acid is D. Okay, so I think you have to chew this as well. Not chew, you have to know this. Yes. What's left? I think now let's talk about the tRNA. So I don't know if there's much. Yes, let's talk about the tRNA. So this is the structure of the tRNA. Okay, it has three loops. It has these sections: the D loop, anticodon loop, and the T omega C. I call it's not an omega, but I call it omega. Okay, T omega C loop, right? And on the anticodon loop, it has the anticodons. Okay, these three bases. Okay, that's what is going to read the codons on the mRNA. Okay, so this shape of the tRNA is called, is it the clover leaf? I've forgotten. I don't know if it's stated over here. It's not stated. So this is the shape of a tRNA, and it has single strand. This is acceptor site. The reason why we call it the acceptor site because this tRNA has to go to a particular enzyme called the amino acyl transferase enzyme. Okay, and that's going to give it an amino acid. So it accepts the amino acid on here. Okay, so the acceptor site accepts the amino acid based on the anticodon sequence over here. Then it will take the amino acid, then it will transfer it into the ribosome, then it will use the anticodon to read the codon and release the amino acid. Okay. Yes, I think that's it. So there are non-protein genes. Okay, some like genes that code for rRNA. It is coding for something, but that the end product of the transcription is not a protein. So not all genes code for a protein in this case. Okay, so ribosomal RNA, I think, well, let me not say we've not discussed it, but they are a major component of the ribosome as well. So the ribosomal RNA or the rRNA is the structural component of the ribosome. Okay, it has the sequence complementarity to regions of the mRNA that actually helps it bind to the Schindler organo in prokaryotes and Kozak sequence in eukaryotes. Okay, and we have other non-protein or non-coding RNAs are the snRNAs called small nuclear RNA. Okay, and this is actually found in the machinery that's used for splicing. Okay, so specifically called as spliceosome. Okay, so let's say it partakes in processing of RNAs, especially mRNAs. Okay, and it can travel between the nucleus and cytoplasm. Sorry. Another example of the non-coding or non-protein genes or RNAs are the SNO, small nuclear RNAs. Okay, this is involved in processing of tRNA, sRNA, sorry, splicing of rRNA. So snRNA is splicing of mRNA, snORNA is splicing of rRNA. Okay, it also does different modifications aside splicing. It also helps in chemical modification of the bases. I want to confirm something. Which a minute. I want to say something, but I might lie. So let me confirm something for you guys real quick. Okay. So yes, it does not the snORNAs. It does not only splice RNAs. It also splice tRNAs as well. That's what I wanted to confirm. Yes. So it is involved in chemical modification of the basis of these RNAs, splice them, and these RNAs are the rRNA and the tRNA, and snRNA is involved with only mRNA. Okay. There's the structure of ribosome. Okay. There are two subunits of a ribosome. In prokaryotes, is this a prokaryote? Okay, let me use this one. In prokaryotes, the whole structure is called a 70S, and in eukaryotes, the whole structure is an 80S. But the individual subunits do not add up to 80 or 70, so don't be confused about this. So in prokaryotes, for example, the 70S, when they separate, are 30 and 50. They should sum up to 80, but it is 70. And in eukaryote, 60 and 40, they should sum up to 100, but when they come together, it's called an 80S. The S is just Svedberg. It's just a unit of measure of size based on how quickly an object sediments in such a centrifuge. Okay, so S is that does not really mean like the size or yeah, that's it. So the bigger one is the larger subunit, and the smaller one is the small subunit, of course. So in the larger, so when the mRNA, let me talk about when the mRNA comes in. When the mRNA comes in, it binds. The mRNA binding site is mostly on the smaller subunit. Okay, and in the smaller subunit, that's where the mRNA comes in, and the mRNA has a sequence. So in the prokaryotes, to be the Shine-Dalgano sequence, when it comes, the mRNA, the small subunit to recognize the Shine-Dalgano sequence and bind to it, right? And in the larger subunits are three tRNA binding sites. Okay, we have the A site, P site, and E site. A is the amino acid tRNA binding site. That means a tRNA that carries an amino group. Okay, and the P site is the peptidal tRNA binding site. P site is where the peptide bond formation, or polypeptide synthesis takes place, and the E site is the exit site. So where the just red tRNA will be pushed there so that it will leave. Okay, then other tRNA is coming. Okay. So if you take a close look, you realize in the ribosome there are rRNAs. And what did we say about rRNA? It is the main structural component of ribosomes. Okay, that's why you are seeing these red things in the ribosome. Okay, so it is the rRNAs that form the ribosomes. Okay, so the ribosomes, you realize, let me use okay, let me talk about let me use the prokaryotes as an example. Realize we have 16S, 23S, and 5S. Okay, so I'm not sure we have those diagrams here, but the rRNA during splicing, okay, splices in different subunits. The small subunit, that's the 5, the intermediate 16, and the larger subunit, that's the 23. Okay, and the 23 and the smallest are found in the larger subunit of the ribosome, and the 16S, which is the intermediate, is found in the smaller subunit of the ribosome. Same thing happens in eukaryotes as well. But in eukaryotes, we have two small subunit, the 5.8 and the 5s. Okay, 5.8 is actually attached, in this case attached to the 28s, which is the largest. 5s is also there, and the intermediate in our case is 18. Okay, punctuation. So as I said, stop codons do not code for any amino acid. So you realize there are codons coding for several amino acid. AUG, which is a start codon, as I said, will always code for methionine. The rest will be coding for their respective amino acid. But when you get to a stop codon, they are non-coding codons, right? And that will be a signal. And if it's a eukaryote, polyase will be added and blah blah blah. Transcription will be terminated, right? So this is the amino acyl transferase, amino acyl tRNA synthetase. I've been saying this the wrong time. So this enzyme is called amino acyl tRNA synthetase. Okay, so it actually harbors the amino acid to be loaded onto the tRNA, and mind you, the tRNA uses its acceptor site to collect or to receive the amino acid from this enzyme. Okay, so when it comes in, based on its anticodon sequence, the amino acyl tRNA synthetase adds the amino acid onto the tRNA. Okay, then it goes away to the ribosome with this, or carrying the amino acid. Okay, so without the amino acid, we see the tRNA is uncharged. Okay, but with the carrying the amino acid, we call it a charged tRNA. Okay, we might not go deep into the initiation and everything. You would have realized the methionine used for initiation is different from the other methionines you are going to find in the protein. Okay, not as in the amino acid itself is different, but how the tRNA is charged is different in both eukaryotes and prokaryotes. But we're not going to talk about that. So what is Wobo hypothesis? So this actually the English is not coming. It contradicts the Watson base pairing rule a little bit. Okay, why? Because there is codon degeneracy when it comes to the codons. Okay, so in that case, the tRNA should be able to accommodate these degeneracies. So in the case of, let's say, serine, was it serine? Oh shit, I have to go back. So in the case of, let's use proline. You see, proline has 4 codons coding for it, CCU. Okay, and we have codon bias as well. So let's see, codon has 4 codon has 4 codons that codes for it. I said codons. Proline has 4 codons that codes for it, CCU. So let's say CCC is actually preferred. Okay, but there are some instances where you find CCU. So because CCC is preferred, the tRNA that comes in is going to be GGG. That's the anticodon. Okay, but whenever the tRNA should know that whenever a CCU comes in, it should consider, right? Because they also code for the same thing. So even though the anticodon is GGG, it can still bind to the CCU. Okay, as if it was GGA. Okay, that's the Wobo hypothesis. So a tRNA that is not exactly matching the codon on the mRNA can still bind to it. Why? Because that particular codon codes for the same amino acid. Okay, that's what I mean by the Wobo hypothesis. I think I've mentioned this. So I think I should just chip this inside. The first tRNA that comes in actually lands at the P site. Okay, so on the ribosome, you realize we had 3 binding sites in the larger subunit. The first tRNA that comes in with the methionine comes here at the P site. Okay, and that's what initiates translation. Okay, but subsequent one that comes in will start from here at the A site. So the first one comes here and bring the amino acid somewhere hanging over here, somewhere around the side. The first one comes in, it joins with the amino acid, then it moves to the exit site and goes away. The next one comes, another one joins at the A site. Okay, we might be through with module 2. I'm sure there's not much to discuss. So of course the bond has to be broken from the tRNA. The amino acid has to be taken off the tRNA so that the tRNA can leave. So yes, that's it. That's it for translation. There's not much to it. So the finished. So after everything, we cleave the peptide from the ribosome. Then they will be transported to wherever they have to go. And I think this one will be discussing it as well later in our discussions. All right. So I'll see you in module 3. Right.